Hi, I'm Vanessa Usoh, and I was an intern at the Palop lab, where I worked with my mentors, Patrick and Stephanie. The title of my presentation is going to be Leveraging Machine Learning to Identify Behavioral Deficits Due to Aberrant Microglial Activation. In many neurodegenerative diseases, such as Alzheimer's disease, there are many seizures that are hard to detect in early stages of disease progression. This is why the motivation for this study is to establish an additional measure of seizure susceptibility in neurodegenerative disease mouse models. In order to model this to the mice, we have used repeated exposure to pilocarpine to understand seizure susceptibility. We're trying to answer these two research questions. How does the transition into disease-associated microglia affect neuronal function? And how can machine learning, such as DipLab-Cut, help to identify behavioral phenotypes in mouse models of neurodegenerative diseases? To answer this question, we used a machine learning tool known as DipLab-Cut, which is a state-of-the-art human pose estimation algorithm that allows us to estimate the pose of a subject and also quantify the video. We started by subjecting these IKK beta CA mice models to a drug exposure model known as pilocarpine, which is a strong model of seizure. And when given in doses to the mice, it shows how frequently they have seizures. Afterward, we collected their videos, and then we subjected them to a frame annotation model using DipLab-Cut, where we labeled the body parts. And then we subjected them to a training with the help of Google Colab Pro, where we created the labeled videos and also came up with the pose estimation for the videos. An example of this can be seen in the video to the right. Here we can see the mice having seizures where it switches its legs back and forth and where it wiggles its tails. We can also see an example of the labeled body parts over here from the nose all the way to the tail base. Using the tracking of that video, we obtained some plots. As we can see to the top left chamber over here, all the body parts are distributed in the chamber, and they're mostly hanging around in the corner, which is typical as the mice are usually hanging around in a corner during seizures. On the bottom left chamber over here, we can see a histogram plot showing just how much the X and Y coordinates are changing. And we expected it to be around 0 because the mice they're not teleported. So the top right chamber over here, we can see the X and Y position over time of all the body parts in the video. We can also see some really great features as we see jitters over here. This is just showing that the tracking is able to pick up when the mice are actually having seizures. To the bottom right chamber plot over here, it tells us the likelihood of each point for each of these points throughout the video. The likelihood are mostly around 0, but we expected them to be around 1. This happened because there's some of the points that have had low likelihood, of which can be improved using just by refining the network. Overall, these results show that we're able to track these mice successfully, and we also came up with conclusions adhering to that. Number one, all animals exposed to pilocarpine have seizures in a dose-dependent manner. And after successfully training and analyzing their notated videos, we actually came up with a pose estimation for this mice, enabling us to track it even more better. Building upon these conclusions, we expect that the next step for this project is to use an even more advanced machine learning tool such as BAME, which is capable of distinguishing running versus walking versus seizures versus turning that were not once possible. And even in the near future, we hope that machine learning such as DipLab-Cut might be used to track humans and help properly diagnose cognitive decline in other neurodegenerative diseases such as dementia. I want to say a big thank you to the Palop Lab for accommodating me throughout this research internship. A big thank you to the sponsors, Suda Emma, the PUMAS grants, and my fellow interns. At this time, I'll be entertaining questions if there are any.
There's a question for you, Vanessa. How many video frames are needed to decide that a seizure is starting versus just a normal movement? Thank you, Robin.
So we actually used there are 25 video frames, 25-minute videos, and how the network shows you how it's stopping versus pausing is that it actually tells you, because it's VAME allows for segmenting and sequencing. So throughout the span of the video, it actually just says it on the screen, oh, walking, turning, and moving.